All right. Again, sorry for the delay here, but here we go. So last time we left off talking about photocatalysis a little bit after kind of going through some of the advanced oxidation processes, some of the more standard ones, and then I told you we'd kind of take a look at some of the, I guess, more innovative, potentially niche or up and coming processes. So that's what we'll do today, spend a little bit of time on quenching by non-target compounds, dissolved organics, stuff like that. And just really kind of flesh out a little bit more about photocatalysts themselves, and then take a look at reactive hydrogen species. So using chlorine as a photo-driven advanced oxidation process, along with singlet oxygen sensitizers. Okay, so to pick up where we left off last time about the photocatalysts, kind of the summary for the key challenges in terms of why photocatalysts really haven't become all that they were promised to be. So back in kind of the 1970s, that was the first time they were really introduced as, hey, this stuff we can use it's like a catalyst, but we can drive it with light and have a photocatalytic reaction and potentially treat water with that or do some interesting things. But 40, 50 years later, we really have no practical applied photocatalysts. Now I think there is one or two systems that are kind of an industrial scale photocatalyst reactor, and to my knowledge, they involve kind of probably a CSTR type slurry. It's a very thick slurry, like grams per liter of TiO2, or some maybe a modified TiO2, and a whole bunch of light added. Of course, that's not going to go very far through the solution, through the slurry, but it's a system that you can put kind of lots of organics in, and it keeps it in there long enough to pretty much mineralize the organics. So there is, I think, one or two systems, but I really don't know if it's actually cost effective, and it certainly hasn't been scaled up to be an industry or a significant part of an industry. So there's a little bit going on, but the real reasons why we haven't seen it become kind of a mainline technology, quantum yields, the light absorption challenges, the stability of the catalytic materials themselves. If it's not some mineral like titanium dioxide or something, then you have some concern about it degrading itself. So that's a concern for many of the photosensitizers, which may be like a dye molecule. You'll have dyes that can sensitize reactive species, but then of course they can also be photo-bleached by the same reactive species. So there's some issues there. Also, if you're going to stick them onto something, for a downside for something like TiO2 is you can't chemically bond it with some other material. You have to do something more like a ceramic process where you heat it really hot and do what's called sintering, and you're gonna get it to stick better and maybe meld into the material you're working with, but it's not going to be a chemical bond there holding it. It could still be brushed off potentially. So you have some issues of that. Are your particles or your materials going to stick onto whatever your anchor support is permanently, or are you going to have it come off eventually? You also have the diffusion length of our reactive species. Last time we talked a little bit about diffusion limited reactions, and for most of our reactive oxygen species, the ROS, most of those are going to be reacting so quickly that they are considered diffusion limited. So the net effect is if we're producing ROS on some surface here, then we've got that reactive species. Well, it's if it's diffusion limited with its reactions with stuff in the water and with the water itself, it's really not going to make it very far before it has disappeared. And so then you have a serious size limitation in terms of how much reactor space is actually being used. So either you put it into a slurry like that one facility I mentioned, or you have to be really creative with how you're providing contact between the water and the surfaces. And to complicate that matter further, if you have quenching with those non-target species, maybe they can compete for the surface, because the surface, like any catalyst, a lot of the action is happening on the catalyst surface. For the photocatalyst, these reactive species can diffuse out, so that's kind of nice, but we're still most of the action is happening kind of at that surface. So if organic matter comes and sticks to the surface, it's essentially fouling it and preventing other stuff from reacting. Okay, so I wanted to, last time I mentioned one of the more creative options I had seen to overcome this was a coated fiber optic cable, and I found, as I was looking yesterday, I found one new example from a couple of years ago where somebody did coat a polymer fiber optic. They used PMMA, polymethyl methacrylate. So that's kind of like our plexiglass type stuff. So using that as the fiber cable, and they actually did put TiO2 on it. So I don't know how long that fiber will last, but perhaps it's long enough. Maybe it's just a proof of concept. But they essentially used an LED source, shining a light here, had some sort of a reflector system to guide the light into the chamber, and then, or into the fiber, and then you'd have the light going through, and then the catalysts loaded on the surface. And you can see from their picture here, you can see kind of probably it's UVA light that they're using there. Yeah, get rid of this stuff. And you can kind of see it. The light source would be here, and it's shining through the coated fiber, and then at some point they have it allowing it to emit through, and that would be exciting the catalyst there. So perhaps you can take a whole bunch of these, like you would in a kind of a hollow fiber filtration membranes, and put them in, have the light shining, then you get a pretty good surface area with no limitations of light going through the water and through whatever's in the water. So that's, to me, I think that's probably, if we do end up with applied photocatalysts, that's going to be likely the way, or similar to the way it would end up occurring. So thought that was pretty cool. So that's what I wanted to say about kind of the limitations. One of the, maybe if I had thought to order this a little better, one of the more common, kind of last time we talked about the more common advanced oxidation processes. Well, Fenton reactions is what I would say is on the common side of things. So this is applied in mass. There's quite a few systems that do use Fenton chemistry. It's pretty well known. It is similar in that we're going to be dealing with hydroxyl radicals and hydrogen peroxide. So kind of the similar types of chemistry that's going to occur, but in this case, it turns out it's a reaction between iron and hydrogen peroxide that's resulting in our radicals that we're interested in. This is also the reason that if you ever get a cut and apply hydrogen peroxide to it, you get lots of fizzing when the peroxide meets the blood, because there's a lot of iron in the blood. So that's actually, you're watching this chemistry happen in real time. And so when this occurs, what we see is, so if we take iron 2, so ferrous iron and hydroxide, that will oxidize the iron to iron 3, ferric. It'll get a hydroxyl radical and some base, OH minus. The ferric iron can also react with hydrogen peroxide, and you end up, you can reduce the iron back, so it's kind of cyclical in nature here, and you get this radical and a proton. So if you have plenty of hydrogen peroxide, this will be looping back and forth in some sense. There's some other reactions that do occur. If we have iron 3 with a superoxide radical anion, that will reduce the ferric iron to ferrous and release an oxygen. So that's probably where the fizzing is coming from, is once you have some superoxide radicals in solution, then this reaction will occur and you'll be creating oxygen gas there. We can also have ferrous iron react with hydroxyl radicals directly. That will essentially quench that radical, get hydroxide and ferric iron. And finally, as we talked about some last time or the time before, we can have peroxide react with hydroxyl radicals to form this guy and water, and that was the one we were taking a look at up here. Okay, so we can take a visualization to capture all of this. That's what this complex kind of source and sink chart is showing us. Essentially, just like the other systems, it is complex. There are sources and sinks of the radicals, and you can see that if you have a set amount of peroxide and iron, these will cycle until all the peroxide has been consumed, and the iron is just going to be going back and forth between ferric and ferrous. So these are called Fenton reactions, where I actually don't know if Fenton might be somebody's name or it could just be related to the root of iron, and I don't know which. But I think of it as Fe is iron, so Fenton kind of makes sense, right? So these are pretty well documented, and you can look at systems in nature where this may be happening. If maybe in an ocean or something, you've got sunlight, you end up with some hydroxyl radicals, and then there will be Fenton chemistry that might be relevant in environmental systems. But we can apply it. Peroxide is relatively cheap and easy to manufacture, so we can apply this, especially if we have some water perhaps that is rich with iron. A lot of times, if you have lots of iron, you're also going to have arsenic. So if you're worried about arsenic removal or interested in performing arsenic removal, then one of the things you might do is add some peroxide. You'll have some Fenton chemistry happening, and these peroxide, essentially the hydroxyl radicals, will also react with things like arsenic to oxidize those. And we usually want that reaction, we want to oxidize the arsenic so that it is easier to precipitate. That's usually the goal for arsenic removal, is get it into the highest oxidation state. Okay, so we could look into the details of this graph, but essentially it's just taking these reactions that we have here and mapping them into kind of where the different chemicals end up and how they loop and cycle. It also shows how methanol or bicarbonate can affect these systems as well. Okay, so the next topic I'd like to introduce is a kind of a newer topic that, in some ways it's been around as long as chlorine has been around for a long time. And if you've ever dealt with chlorine, you probably know that if you leave chlorine in the sunlight, it's going to degrade. It's going to that will drive off the chlorine. And actually, there's a fair amount of relevance here to swimming pools. If you have an outdoor swimming pool open to the sun, the sun will be slowly degrading the chlorine, and you might not have thought about it, but you're actually probably producing radicals during that process. So it's actually a little bit of photolysis going on there. So this is a project that my research group is embarking on in some sense, is using new LEDs to enact chlorine photolysis. So one of the reasons we haven't used chlorine as a advanced oxidant in the past has in part been the fact that our traditional UV lamps, UV 254s here, if we match them up, which these wavelength axes are matched, you'll see that it excites HOCl fairly well, but OCl minus absorbs a lot more strongly in a relative sense. Now, there are some quantum yield differences. It turns out if you use the same amount of photons here compared to the same amount here, HOCl does have a better quantum yield, but you're getting better absorbance over here, so there is some balance there. And it looks like perhaps the optimal might actually be somewhere around a 265 range. So you're getting a little bit of both. You can potentially control the pH to control which, if you did want more OCl minus, then you can use basic conditions. If you wanted the HOCl, of course, you can do acidic. So there's adding the different wavelengths in here gives us another avenue to control the system that has not been present in the past. So I've got a little box full of different reactions that can occur here. And if we take a look, a few highlights here, well, of course, bleach or the chlorine itself, the free chlorine is important. We can also potentially use chloramines. So monochloramine and dichloramine here. And what will end up forming is chlorine radical, so a Cl dot just by itself, a Cl2 dot minus, so I don't know if this has a name, but essentially a Cl2 radical. We will end up with some oxygen radicals, O dot, or O dot minus. Some hydroxyl radicals can be formed if this O dot minus is formed. And we can also get ozone out of the system in some cases if we were to follow these reactions. So if we get this O dot radical, and I think that minus actually might not, I don't think that should be there. We talked about these radicals or these that situation last time where we had that O1D or the O3P. So these two products here are reactions are formed when we have OCl minus going through photolysis. So those will essentially split that OCl minus into Cl minus and O dot. And that can be either in that O1D or O3P. And that will determine whether or not we get essentially ozone formation by reaction with that O3P, or we can get hydroxyl radicals with the O1D and water. So those two reactions, we can get either ozone or we can get hydroxyl radicals. So it's kind of interesting. We can end up selecting based on the solution conditions because one thing that this depends on is pH. So we can adjust the pH or adjust the wavelength, and we can kind of select for one or the other there. So the quantum yields are obviously important. And again, the new lights are providing us some control over what we have in our system and what reactions we are driving. And another cool thing that we can do with LEDs is actually switch them on and off instantaneously. And with more colors or more wavelengths we can use, and that ability, there is potential to switch back and forth between different wavelengths so that we can end up causing a very specific set of radicals, and perhaps even this might be the wrong class. I don't know.
I'm not here. I'm good for Dr. Arizani. Show up at this room at 9:30.
Okay. Well, I'll be teaching until 10:20. In case, so you're welcome to be in the lecture if you want. But this, okay. So the radicals then, if we switch back and forth between wavelengths at a given pH, we may be able to cross back and forth between these types of reactions so that we are generating some ozone and then selectively either photolyzing that ozone or letting that ozone kind of exist for some time. So we may have some control at more of a very fast time scale and potentially ramp up a particular type of oxidant that we're interested in for the reaction. So that's something that I'm curious about, whether or not we'll find any real potential application there from that new capability we have. When we know that the lower wavelengths, we tend to get the less selective radicals, the hydroxyl radicals, and to a lesser extent the chlorine radicals, which they are generically non-selective, but they have, I guess they're more selective than hydroxyls. So if we have complex waters with lots of organics, maybe we want to set up the situation where we're not getting hydroxyls. Perhaps we can get only chlorine, or perhaps we can set it up to just do the ozone. And perhaps it would even make sense to change this based on the solution conditions. So as water's coming in, the water quality changes, maybe it rains, then perhaps the system automatically switches to a different radical. So some interesting ideas there that we'll be taking a look at. Another kind of newer advanced oxidation process is persulfate oxidation. So persulfate systems is actually one of the only radical platforms, I'll say, that does not have hydroxyl radicals as a major part of the process. So we're not going to end up forming hydroxyl radicals. There's another piece of art, again from my PhD advisor, another great, and this one was a review paper that he published with a couple of his colleagues a year or two ago. Yeah, it was last year. And well, I actually took a pretty good look at this paper as I was preparing a proposal. I didn't end up going with the persulfate route, but it was in some way comparison to the reactive halogen type stuff. So it was an excellent review that included peroxy monosulfate, which kind of funnily has a PMS as its acronym, and peroxy disulfate. So this is essentially sulfate radical or the disulfate radical. And so they were comparing, okay, what types of roadblocks and obstacles are on this type of these type of reactions face, and which one would be better. And so this nice little picture is showing the different things along the way. Maybe singlet oxygen plays a role sometimes. Have a little single oxygen thing there. The roadblocks of natural organic matter or bicarbonate. Chloride ions can also react and interfere. And so the question is, so in one case I think one of them you will form the hydroxyl radicals, but the other is just the sulfate radicals. And so that's, given that those are our endpoints in question and what we're considering, which one's better to use, and how do we control it? What are all the reactions going on? So that's this is the persulfate oxidation is one that's could very well become kind of a widely applied. It is a technology that is fairly mature. We know a lot about it. And the sulfate does provide us a kind of a different avenue for a reactive species than our other systems. Another neat thing that they did in that paper was kind of collect data from all sorts of other papers that they were reviewing and plot the reaction rate constants that had been observed for all these different papers for different types of molecules. So here they're taking a look at the first few are alcohols and acids. So you've got like ethanol, methanol, allyl alcohol, terbutanol, fumarate, and ascorbate, all these different things. And so what they did was they took a look at the second order reaction rate constants, so in per moles per second, and they plotted those for each of the oxidant species here. So ozone, we have a pretty low for whatever this 11 is, acetate, and you get higher reaction rates for some of the other ones, right? So ozone has that range of different molecules for that. Singlet oxygen, this is the excited state molecular oxygen, can react with several of them, and generally a little bit faster than ozone in most cases. Then we have the sulfate radical and hydroxyl radical. So there's certainly other radicals that you could potentially do this type of thing with if you had chlorine radicals, for example, superoxide radicals. There's a lot you could potentially consider here, but I thought this was a very neat way to represent the speed at which these different oxidants are reacting. So you see sulfate is actually pretty close to hydroxyl in terms of reactivity, not quite as fast. Singlet oxygen would be next, and then ozone last. Ozone's not truly a radical, so it kind of makes sense. So here you can kind of see this is true for alcohols and acids. Also seems to be pretty much true for aromatics. And then they've got another couple. So some inorganic anions like azide, bromide, chloride, iodide, bicarbonate, carbonate, cyanide. So several different inorganics. And in this case, ozone actually spans quite a large range. So 10 sulfite, for example, reacts pretty quickly with ozone, as does iodide. So lots of these have pretty quick reaction rates there. And then some other miscellaneous ones also listed. And you can kind of get a feel for the reactivity here. So that's mostly the reason I wanted to show you that, was just kind of a comparison to kind of put all of these into perspective, how the radicals are comparing with one another and how variable are they given different targets. One thing you'll note is that the hydroxyl radicals are just really pretty well reactive with almost anything. On both of these figures, the lowest for these two was 10 to the 6th, and then there was one that was around 10 to the 4th here. That was number 8, so that was hydrogen phosphate, apparently. So anyway, I thought that was kind of a neat set of information there about radicals. Okay, so next I want to tell you about singlet oxygen's photosensitization. This was really my PhD topic, and so I could spend hours boring you with just this if you wanted, or if I wanted, I suppose. But I'll keep it, on the brief side. Essentially, the photosensitization of singlet oxygen from, and you'll see in a moment, I have the animation, we're essentially going from triplet O2, which is the ground state, to the singlet state. Oxygen is just kind of weird, the way its molecular orbitals work. It's weird in that most times in the ground state, things are in a singlet state. But diatomic oxygen is a weird one, and it is in a triplet state. So the excited state for oxygen then is a singlet. And that means it's pretty reactive, and we can use it, as we just saw, to degrade certain compounds if we've got some single oxygen around. So it's essentially the same goals as the semiconductor photocatalysts in terms of using light producing something reactive and applying that reactive species. So essentially what you do is you excite some molecule. That molecule then gives its excitation to the oxygen, and then that oxygen is going to be our disinfectant, or it's going to be the thing that we're interested in because it's toxic to the organism. This is something that happens in biological systems all the time. So a lot of single oxygen research is just trying to understand its role in kind of biomolecular kind of context. If we're exposed to the sunlight, or if cells are producing single oxygen in some other way as a trigger to for cell death or something. It's this is kind of what we look at and what we're interested in. So a fair bit of research in that regard. In terms of a disinfectant or a tool, it can be better than our hydroxyl radicals in that sense that it reacts slower, right? So if you take a look at the single oxygen here, generally it has more selective reactions. In some cases it reacts pretty quick, but most of the time hydroxyls are consumed a lot faster, they're reacting faster compared to the single oxygen, right? So and we can take a look, maybe they're faster with some molecules than others, but essentially that's a potential advantage if it's still effective enough against our viruses or whatever we are trying to degrade. Okay, so a little bit more technical, the way it will work, and I'm using C60. This is a Buckminster fullerene as an example. This again coming from my work as a grad student back in the day. The way it works is we take a ground state, and this is true of almost any photochemical reaction involving excited states. So what we're going to see here is something called a Jablonski diagram. It's just looking at energy level and tracking where the molecular states are. So with the light, we excite the fullerene. In this case, it's then in a singlet excited state. So we've just taken one electron from inside the fullerene and knocked it up in energy level. It hasn't gone anywhere, but it's excited now, and that's what this star is denoting. And we can actually know something about how much energy that has. And it takes this gap between for a standard fullerene. This is we mentioned oxygen allotropes the other day. This is a carbon allotrope. So it's just all carbon. There's a few different conformations. You can have C70, C60. There's a couple that are smaller, but mostly larger. And C60 is kind of the most common and most basic form. It's also exactly the orientation that you would see if you looked at a soccer ball, a typical soccer ball with the hexagons and pentagons. So if you were to count the vertices on a soccer ball, you'd find 60 vertices, and it would be that same shape. So this energy level that's required, you'll have to have enough energy in your photon to reach that excited state, and this is 1.9 electrovolt electron EV, electron volts. That corresponds to light that's at least 650 nanometers. I guess at most, right? So the highest wavelength, the lowest energy light you can give it to achieve that is 650 nanometers. Now, most of the time for these systems, what you're going to have is an excitation that comes somewhere above it, and then anything in this range where you reach that, it will then relax down to that level and that stable level there. So usually if you did want to excite this, you're going to want something that's a little bit more energetic than that 650, because if you remember kind of basic chemistry, the activation energy plays a part. So you have to have some help to go over an activation energy barrier things like random motion, heat, can help that. So in this case, what you would want to do is shine light that's more wavelength than that, because sometimes it won't absorb or it won't achieve the excitation like you were wanting if you were just doing a 650 laser. Okay, so in that said, if we were to look at the UV absorption spectra for fullerenes, they absorb pretty strongly in the UV range, a little bit in the visible range, but you can kind of modify that a little bit if you add attachments. Okay, so then what happens is that singlet excited state in some molecules, in particular molecules that are good at photosensitizing singlet oxygen, essentially it will go through an internal conversion process. Normally this is spin limited, or spin disallowed, to go from a singlet state to a triplet state. But the nature of this molecule, of the C60, the fact that it's kind of bent actually helps that spin transition happen. And this is called intersystem crossing. It happens that fullerenes are very efficient at this, so almost all of the singlet state C60 transforms into a triplet state, loses a little bit of energy doing so, just vibrationally relaxing, and then we're in a triplet excited state. And that is 1.5 electron volts above the ground state at that point. From here, this is the type of molecule, or this is the type of situation where you might have phosphorescence. So fluorescence is when you have that excitation and then it relaxes down maybe to here, and then you have a molecule that kicks off another photon at a lower energy level, and so you absorbed maybe a blue light and now you're emitting a red light, right? So that's typical fluorescence, and it happens pretty much immediately. As soon as it's absorbing, it relaxes and releases a photon, and then the ground state, and then it relaxes back down to ground state. That would be a fluorescence process. And if you've ever played with a UV light or something and looked at different things, if you have a white shirt on or something, sometimes it'll kind of glow a blue under a UV. I was actually doing a demonstration for middle school students just a few weeks ago, and we used a laser pen, one of the violet laser pens, and you can use it with like food coloring and different things, and you can actually have pretty good fluorescence with several different food colorings and random like household liquids around the house. So that's fluorescence. Now, if you have a molecule that goes through this intersystem crossing process and you end up with a triplet state, those typically live or stay there a little bit longer and are a little more stable, because that transition from the triplet back down to ground, then you're going from triplet to singlet, that's disallowed in terms of the spin, electron spin stuff. So it tends to hold it longer there, and then occasionally they can spit out a photon when the conditions are right, and that would be phosphorescence. And that's the glow in the dark stuff. So if you've ever played with anything that's glow in the dark, you hold it up to the light, let it absorb lots of photons. Essentially what you're doing is you're getting it to here and letting those relax. And then when you take it into the dark place for some time, until it's exhausted those excited molecules, you're getting some molecules kicking out here. And so just like in the fluorescence case, you're going to expect that your energy level of the photon coming back out is lower than what you put in. So if you had a glow in the dark thing and you knew it was going to glow green, and you tried to excite it with a red light, that's not going to work. I've never tried this, but just based on this principle, it shouldn't. Now I'm thinking about how can I try this someday. Red laser pen versus a violet laser pen or something. Might actually be a good activity for middle schoolers sometime. Okay, so that's kind of the typical photochemical processes that we can sort of relate to. The other thing that can happen with this triplet state, and which does happen with fullerenes don't tend to phosphoresce. They just it just doesn't happen. Some molecules are better at it than others. But what will happen is if there's oxygen nearby, it is pretty effective at transferring that energy to oxygen, and then you have singlet oxygen, and the I guess you could call it the redox potential, but the energy level of singlet oxygen is at 0.98 electron volts. That corresponds to a photon of, the photon energy of photons in the infrared range. You can actually see, you can detect singlet oxygen by using infrared light and just checking the absorbance of light in that range. And you can detect a signal there for singlet oxygen. So anyway, that is an oxygen species that is more reactive, and you can see it's, these transitions, even if you used a pretty energetic photon that was required to initiate it, the end product, you have energy losses along the way. So that's one source of inefficiency if you're looking at just the amount of energy put into the system, because every time you have that relaxation down to the next step, it might go like that, and your excitation might go like that. Each time that happens, you're losing a little bit to heat. It's just a vibrational relaxation. And some of the times, if there wasn't an oxygen around or whatever, it could just relax vibrationally as well. But for fullerenes, if they are in the right condition, completely dispersed, then this happens pretty efficiently. Now, the project that I was working on, there was a couple things. One was the fact that we can add attachments here to the fullerene molecule without corrupting its ability to perform this process. In fact, you might be able to even enhance it, because you can tweak these energy levels a little bit if you functionalize the fullerene. So then you have a little bit higher or lower energy level here. Maybe you can use better visible light for the process, and likewise higher or energy level here. You just have to balance to make sure, if you are trying to go a little bit lower here to get make more use of visible light, because normally, more photons in the visible range will work if this is a lower energy threshold. You don't want it to also cause this to go up too high to the point where you're no longer able to do the inter-system crossing. It's no longer an energy favored direction, right? So there is some balance there that we were taking a look at in terms of how many functional groups were we adding onto this molecule. The other advantage here is, it turns out you can add several and it usually doesn't matter too much, but it's just kind of something that is adjusted a little bit. But the other good thing here is you can actually chemically bond this to some other material, maybe something that you were anchoring your photosensitizer system with, like the fiber optic cables. You could chemically bond it to them, and then you have a permanent attachment. You're not worried about losing your fullerenes over time into the water that you're treating, because you probably don't want to drink the stuff. I mean, chances are it's probably not too hazardous, but you don't want to do that. You don't want to lose your material if you're trying to use it. So that chemical bond without disrupting the system is a pretty good advantage. The other thing it can do is it can help prevent a common problem when you put a all carbon molecule into water. What normally happens is they stick together, because they are generally hydrophobic. If you think about carbon, it's very neutral. There's just no charge, right? A fullerene, with no hydrogens on it, this is all carbon-carbon bonds that are, we think of like a benzene, right? We have it like that, but a benzene has a hydrogen on each of these corners. Instead of hydrogens, we've bonded it with carbons and repeated that. So the whole thing consists of these resonating electrons, shared bonds, and so the charge really is spread uniformly, and there's just pretty much zero charge across the whole thing. so that they are very prone to sticking together when you put them in water. And it turns out that kind of kills their ability to act as a photocatalyst or a photosensitizer. So part of this whole project that I worked on was, okay, well, can we add the right kind of functional groups so that they don't do that so much, or they can kind of have a little more space between each other and have this photosensitization capability. So we were able to do that, and we ended up using fullerenes with 3 of these bonds there. That was enough to get the single oxygen process to work okay. There's some interesting reasons that worked. But from here, what I wanted to show you is then when you put them in water, they'll still aggregate a little bit, but they're not such tight crystalline clusters there. What will happen is you'll have, if you put it in water and you add viruses and track how many viruses are living over time given some different conditions, well, what we see is we can use a very small amount of those fullerenes, even down to kind of the nanomolar range with the right light source, and get very rapid killing of the viruses or inactivation. So viruses are not very well protected, and single oxygen will attack different components of them. And so just a few single oxygen reactions and the virus is essentially dead or incapable of replicating. So here we're just varying the amount, and then when we got down to a really, really low amounts of fullerene, 10 nanomolars here, we ended up having to use UVA light, and then we got a little bit of inactivation, but it looks like that small amount of fullerenes were eventually kind of fouled with dead viruses or something, so they no longer were working. So all that to say, we can see here an example of quite effective disinfection using this process. But this was in a bulk system. We just put some of these in a bulk, a batch reactor, stirred. And so this is not showing that it is practical yet, because we have not attached it to something and caused this to happen and had this 5 logs reduction of the viruses in a system that we can run continuously, right? This is in a system where the fullerenes are added to the water and are now a contaminant, right? That's again highlighting the problem with this type of technology. There is reason to be excited about it, sure, but there's also reason to be, okay, well, is this going to work? So that's kind of a constant struggle for this type of technology. On the flip side, in sunlight, it's really effective. And when we took a look, I actually took it outside and did it. Even with some natural organic matter, we were still able to have pretty good inactivation, despite the presence of organics, and in a lot larger of a quantity. So 5 milligrams per liter. I think we used Suwannee River natural organic matter versus 250 nanomolar of the fullerene stuff, which that would correspond to probably microgram per liter amounts. So probably somewhere between 100 and 1,000 times less mass of fullerenes compared to the organic matter there. Okay, so speaking of quenching, a few inorganic contaminants. We've mentioned a couple times, we've looked at some of the sources and sinks type plots, and every time they'll show us the bicarbonate. So for advanced oxidation in general, I think single oxygen is usually not too reactive with inorganics. So we did see a few in the slides up there, but particularly for anything producing hydroxyl radicals, and sulfate had its own issues, and so the sulfate radicals from the persulfate oxidation, that review took a look at that comparison between hydroxyls and sulfate radicals for these different inorganic species as part of that review. So there's some important differences there, where hydroxyl radicals, essentially you can oxidize your inorganic molecule or constituent and get hydroxide. So sulfate can do the same thing, but is not going to be as susceptible to, say, bicarbonate here. So bicarbonate, the reaction is that you end up getting a bicarbonate radical, and the hydroxide that can then that'll be in equilibrium with a carbonate and water, and essentially that carbonate, although it may be, in either form, while it may be somewhat reactive, it is not nearly as reactive, and there is no route back to a hydroxyl or anything that can form a hydroxyl. So that is effectively a sink, and it's not going to perform nearly as well as the hydroxyl for oxidizing things. So essentially, it's at that point, for the purpose of the advanced oxidation system, it's considered kind of useless. So it is a route that consumes hydroxyls and does not give us much benefit. Same thing can happen with other weak bases like phosphoric acid, dibasic here, can react and get that hydroxide, and then you're left with a phosphate radical that is not particularly useful. There's a few other ones that can be important, chloride and sulfite in particular. So those two can quench. Likewise, you can just have hydroxyl radicals reacting with themselves to form peroxide. Now, peroxide we can split again and get something useful out of it, but it's kind of a back reaction that you don't really want to have. And the other thing that can happen is you can react hydroxide with two hydroxyl radicals and end up getting oxygen and water. So again, at the end of the day, there's so many reactions that are occurring that you have ways in which you'll end up with exactly the opposite of what you want, and you'll have kind of nothing left over. Okay. With that, I want to go into dissolved organics, and this is kind of an interesting topic that I got into right as I was starting here. And really what we were looking at is, we mentioned all these different compounds can react with hydroxyl radicals, for example. And then if you think about what's in organic matter generically, if you had like natural organics or organics in a wastewater treatment plant, then you're going to have a system where you've got all sorts of different compounds there and large molecules that consist of all sorts of different molecules. And one thing we can do is sort these based on their fluorescence signal. So we actually introduced fluorescence a few minutes ago and talked about that, how that works. So you can do these neat three-dimensional, so to speak, images where you're looking at, the X and the Y here are the emission wavelength of your fluorescent signal, so the photon that's being kicked back out from an excited state, versus your excitation, so what wavelength you use to excite it. Different molecules are going to absorb at different ranges, and they're, if they can fluoresce, not all molecules fluoresce, but if they can, so molecules that contain what we call fluorophores, so just compounds, groups that can fluoresce, maybe that one can or something, or maybe the benzene group can. Whatever the case, what we're looking at is you can know something about the molecule if they are excited by a certain wavelength and they emit at a certain wavelength. So it's a very subjective type of analysis, but in general, we've recognized that you can classify different different types of macromolecules, stuff in the generic natural organics, based on their characteristic signal with this excitation versus emission. Now, the third dimension here is just the fluorescence intensity. So the more intense the color here, and don't worry about these signals. These are just artifacts from like water. But the more intense the signals here, the more molecules there are that are fluorescing in that region. That's what that means. So that's why they call it three-dimensional excitation emission matrix here. So if they are excited in the low UV range and also emit in that low UV range, those often correspond to aromatic proteins. So proteins with a lot of aromatic compounds in them. If they are excited, again, in that low UV range, but their emission is kind of pushed further to lower energy, we've generally classified those as fulvic-like molecules. So fulvic acid is, we have these terms like fulvic acid, humic acid, and then there's also tannic acid. These generally relate to what type of material these organic acids are coming from. Humic acid is not just one molecule, it's a collection of some. Tannic maybe is just one in particular, but there's similar ones like it. And tannins, if you've seen any of the waterways in Louisiana, chances are it's got a brown stain to it. That brown stain would be from degrading leaf matter. That's tannic acid primarily, or tannins generally. There are some that the biological activity remove a lot, but then humic acids are related to soil type materials generally. So the way we even talk about those different classifications is also a little bit subjective. So this analysis is interesting and a little bit useful, but generally kind of subjective, right? We don't know exactly what these are about, but we know if we take a look at the fluorescent signal from soil type organics, generally it's in this category. That's kind of the picture here. The last one is, or no, not the last one, but number 4 here is SMP. That's soluble microbial products. So we've got bacteria. They are eating stuff and producing soluble waste. So I guess bacteria excrement maybe, or bacteria, defense mechanism molecules, whatever they're going to produce. They're known to produce stuff. So especially in like a wastewater treatment plant, whatever soluble molecules that they are putting out, those are generally classified here. So if you were to take a look at wastewater treatment and you get a big signal there, that's the type that you think that we're looking at. And then humic acid over here in that 5th category. So the point is, what we want to do is be able to have some idea of how reactive these guys are going to be with their different radicals. And the reason we want a tool like this, that maybe we can know that one section or the other is going to be a problem for hydroxyl radicals, but maybe not for single oxygen. The reason really is because there are so many different molecules that exist in nature, in these organic matter samples. So here's a couple graphs from some paper. I apologize if I didn't grab the citation with it. I meant to. But this is a paper that looked at a whole bunch of other papers that were essentially building a map of the carbon to hydrogen ratios. So I think this is on the axis here, this is hydrogen, how many hydrogens divided by carbons versus oxygens divided by carbons on the x-axis. So basically plotting unique molecule types or unique compositions of hydrogen versus carbons versus oxygen versus carbons, just a million different molecules, and each of them are going to have different reactivities, right? And so how do we even approach that type of system when there are 3,000 distinct formulas on the left one and 2,500 on the other one. It's just an impossible task, right? And also to know whether or not, which ones are in your system, and to know then, okay, what types of reactions are happening based on these, functional groups that are there. So that's kind of the reason we'd want something like that 3D EEM analysis. And so that's what we did. We took an analysis and compared a few different fractions. If we took some organic matter from a wastewater treatment plant, fractionated it, which is something that our colleagues in France did, and then took a look at, okay, well, maybe we can separate it into like a hydrophobic and a hydrophilic and like a transphilic. So we've got hydrophobic and a transphilic, and then kind of just whatever colloids were around, and also compared it with humic acid. So we looked at the reaction rate constants between, what we observed between degrading this molecule in competition with all these different other organic matter fractions that we used that 3D stuff to characterize. So we know something about the molecules, and we applied, maybe we separated the colloids with some sort of filtration. We used some sort of ion exchange resin to get the hydrophobic type of molecules, and we took a look at those. And so with the combination of these two techniques, what we were looking at is, can we separate and see a difference in the type of quenching that happens? And it turns out you can. For example, this humic acid quenched the process pretty quickly compared to some of these other, the hydrophobic and the transphilic both, because this is just water here on the left. And then you start adding some organic matter more and more as we go, and we see that with more and more organic matter, our rate of degradation decreases, but for some of them it decreased quite rapidly. So that was kind of the point, and we actually identify that these ones that are really rapid, those are probably quenching on the surface rather than quenching just out in solution, quenching the radicals in that manner. And that was building on someone else's previous work. We also took a look at, can we, how much of a difference does this make if we're using like a membrane bioreactor? So a real system that is separating the carbon or the organics in some way. So if we have an operational membrane bioreactor, we got all this organic matter and made fractions from the membrane bioreactor's activated sludge system, then we can compare that same effect if, instead of using like our fancy separation tools to get the hydrophobic and whatever else, instead of doing that, we just let it go through the membrane in the membrane bioreactor, and then see how it performs if the membrane is like just cleaned versus if it's like super fouled and it hasn't been cleaned in a couple months yet, or like chemically cleaned, or somewhere in between. And it turns out there was a pretty big difference. So the really dirty membrane was keeping out more organics, and it seemed like it was organics that were like that colloids fraction. So it seemed like with a very dirty membrane, it was performing better in terms of organic material removal, obviously at a higher cost because you have to put more pressure. But that fouled membrane here, if we take a look, that's this guy in the blue. That one had the least drop in reaction rates out of any of them. And so it was kind of neat being able to see that, okay, well, this membrane, because it's fouled, it's achieving a lot more separation, and it's actually getting rid of the stuff that's causing the TiO2 system a problem. So in terms of kind of practical implications here, it might be best, if we're going to do some sort of advanced oxidation after wastewater treatment, it might be good to use a membrane and let it stay fairly fouled, or design a membrane that's going to kind of remove the same stuff this one is removing. Ideally, you'd remove that stuff without too much energy cost. You can also clearly see that the washed membrane, this is not quite linear. It goes from 0 to 0.5 to 0.5, then then it's linear, but it essentially drops quite rapidly for that case, because that's these bars here. So the cleaned membrane is letting a lot of organics through, but it's not just organics, right? Because this is actually a concentration basis here. So this is not, it's not that it's a different amount of organics, it's the type of organics that it's letting through. And so on a mass basis, that type of organics was causing a big problem to our system. So it was pretty interesting to see that, just in a operational system, just the amount of time since a thorough chemical cleaning on the membrane made that big of an impact. And we kind of were able to predict it based on the analysis we did comparing these 3D graphs for each of them. Apparently I didn't include that in the slides, but we could take a look, and for each sample we have a different characteristic thing here, and it turns out the colloid fraction had a lot of these guys, and the aromatics we know are pretty reactive with hydroxyls. It might also be the case that they were not just reactive but also sticky, so they stuck onto the TiO2 more so than the other parts. And so we kind of were able to see that there was a pretty big signal in this range for the cleaned membrane, which was giving us the problem organics, and for the colloids, which was out of the fractions, that was the one that caused more problem. So that was all I thought pretty interesting. Really, I guess what I should be showing you is my dog pictures, right? I might be able to find one. Let me see. I have one on my phone, but I think I posted it on. Just give me a moment. I think I can get a picture on Discord.
Did you get a dog?
Yesterday. Yeah. This is why I was late today, so I was telling everybody. So that's the doggo. This is a motion shot. I think I've got one more picture. Let's see. It's like a Great Pyrenees and maybe
Lab.
Yeah, it's some sort of lab probably. It was we're adopting, and it apparently grew up kind of in the woods. It was found near like a hunting camp or something, and people would just drop a bag of feed every week. But it
That looks like a long-haired retriever.
It's also part Great Pyrenees because it's got like little I guess they're called dew claws or something on it.
Double dews.
Yeah,
Okay.
and so it's like a lab Pyrenees mix of some sort. So we're pretty excited, except that on the way out this morning, it had a little accident.
Oh,
So
or her.
he's about a year and a half or something.
Mhm.
Yeah. So we're excited.
Now you got the space for it.
Yeah.
Yeah.
All right. So that's all for today. And that's all the pictures I have too. So,
Just dead.
so have a good weekend. We'll see you on Tuesday.